Oh, we've talked about basic Punnett squares, right? So maybe we have some trait, and we have two parents that are heterozygous for that trait, and we know what this Punnett square will look like, right? Parent A, parent number 1, I'll say the male parent. It doesn't really matter which side you put male and female on. The male parent can make two kinds of sperm cells. That's what these little things represent here, are the kinds of sperm cells that they can make. It can either have the dominant gene or the recessive gene. Female parent, same thing. And you guys know that there's the homozygous dominant genotype, the homozygous recessive genotype, and then you have the heterozygous genotype. And that gives you a 1 to 2 to 1 genotypic ratio. But the phenotypes are determined by the dominant allele. So you would get whatever the dominant trait is here, dominant here, and dominant here, because all of them have at least one dominant allele, and then recessive here. So the phenotypic ratio would be different. It would be 3 to 1. Now that's true for a monohybrid cross, which is what this is, monohybrid cross. But something Mendel became interested in is what happens if we have a dihybrid cross. What would it look like in that situation? So let's say we have two traits. We have two traits, and the parents are heterozygous for both traits. So we have the one that we just looked at, parent number 1, is heterozygous for trait A, for gene A, and they're heterozygous for gene Q. Parent number 2 is heterozygous for gene A and heterozygous for gene Q as well. Well, now there's more than 2 types of gametes that they can make. Both of these can make 4 different types of sperm or egg cells. For the male side, wow, that was bad. There we go. For the male side, the 4 types of sperm cells that he could make are capital A, capital Q, capital A, lowercase q, lowercase a, capital Q, and lowercase a, lowercase q. And the same is true because both of these individuals are heterozygous for both genes. The same is going to be true for the mother as well. Capital A, capital Q, capital A, lowercase q, lowercase a, capital Q, and lowercase a, lowercase q. Gives us a 16 square Punnett square. Now, if you want to go through and in order to really confirm this idea for yourself, you can go through and fill out every single genotype here. So, for example, in this top box, you would get an individual that is homozygous dominant for both of these traits. In this case, you would get someone down here that is homozygous recessive for both of those traits, and you'll get lots of different combinations in between. I'm going to fill this out a little bit differently, though. If you want to see a picture of it all filled out, there's one on the slides. There's, let me see here. Right here, this is the same kind of problem with all of the genotypes listed out. But what if we only worry about the phenotypes for right now? Why is that? There we go. Okay, so let's say that we what are these genes going to be for? Let's say that A is tall and Q is, quiet. It can be any kind of thing that we like, so we'll say that the dominant genes code for tall and quiet, just because it's a Q and that's the first thing that came to my head, and it's the morning, so give me a break. Well, any individual with this dominant gene is going to be tall, right? So what do we got? Tall here. Tall. In fact, anyone in this first column is going to get this dominant allele, right? So all of these are going to be tall in terms of phenotype. And same thing with this column. All these are going to be tall. And same thing for these two rows here. All of these are going to be tall. I think it's a combination of the fact that I'm running this on Photoshop so that it doesn't accidentally disappear on me, and the fact that we're running the conference app is just too much for my computer to handle. So if you see me skip a letter, it's not my fault. It's the computer lagging. Okay. Now, what about, so everything else is not going to get one of those dominant genes, so be short. Hopefully that makes sense. Right? We have a 16 square Punnet square, but if you're only worried about the phenotype, you could just go through and write in how many of them are going to have the dominant phenotype, and everyone else is going to have the recessive phenotype for gene A. Right? So we've just covered all the phenotypes for as far as gene A is concerned in this cross. Now for gene Q, I said what? Quiet? Okay, so anything that gets a dominant gene, anything that gets a dominant allele is going to be quiet, so we'll say quiet. How about I do it this way? Okay. So this one will be quiet. This one will be quiet. This one would be quiet. Everything in this column, because you're getting a capital Q going down the line. And also, everything in this top row would be quiet. But also, over here, third column, that's all going to get that dominant allele. Okay, and also, third row down, everything here is going to get that dominant allele. This one already has one. That one already has one. Well, what does that mean? That covers all of our bases, but everything left, there's 4 of these left. It's, I guess loud would be the other phenotype, right? That would be the recessive phenotype in this case. Loud. So what does that leave us with? Well, if we were counting up all possible phenotypic combinations, right? Tall plus quiet. How many of those do we have? 1, 2, 3, 4, 5, 6, 7, 8. Oh, sorry, counted a loud one there. So 6, 7, 8, 9. 9 of those. How many tall plus loud? One tall loud? A little bit harder to find them now. Two tall loud? Ah, there we go. There's a third one, so that's number 8 over here. 3 tall loud. So there's 3 tall loud total. How many? We'll do it in the same order. How many short, quiet? Short and quiet, this would be this one, and this one, so there's 3 of them. And then how many short and loud? One. It's this one right here. Okay, so this is where we get a 9 to 3 to 3 to 1 phenotypic ratio. Just looking at their phenotypes, not their genotype. The genotypic ratio is really, really long and complicated. I have in the past offered as an extra credit question on some of these tests, but I don't usually ask about the genotypic ratio of a dihybrid cross like this. This is a dihybrid cross. Interesting. Dihybrid cross. Dihybrid because these are heterozygous parents for 2 different traits. Okay, so 9 to 3 to 3 to 1 is the normal ratio you would find. That is 9 out of 16, 9/16ths, right? 9 out of 16 of them are going to have both dominant phenotypes. 3 out of 16 will have the first dominant phenotype and the second will be a recessive phenotype. Another 3 will have it flipped. The first one will be a recessive phenotype and the second one will be the dominant phenotype. And then 1 out of 16 will have both recessive. That's the one that got this egg cell and this sperm cell. So when they fertilized, you got short and loud here. That's your 1 in that case. Now, sometimes these problems can actually be simple. I stumbled over my words. Simplified a little bit. So let's give another example. Switch colors up just to keep it exciting. Let's say that instead I have a parent who is heterozygous for 2 different traits. I'll use Q again. I really like Q because if you use Q, there's no question about which letter is uppercase and which letter is lowercase. And the other parent is homozygous recessive for both of these. Okay, this can actually be a much simpler Punnett square. This first parent here can make 4 different kinds of gametes, just like before. I'll keep it on the same sides here. So let's say that this is the male. They can make capital Y, capital Q. They can make capital Y, lowercase q. They can make lowercase y, capital Q. And they can make lowercase y, lowercase q. Those are the 4 different kinds of sperm cells this parent can possibly make. Now, ordinarily, you would think, okay, well, we're going to do another 16 square Punnett square. But the reality is these Punnett squares are only mapping out how many kinds of sperm or egg cells a individual can make. And this parent, this second parent, the female here, female, she can only make 1 kind of egg cell. She has no choice but to make egg cells that are recessive Y and recessive Q. So this doesn't become a 16 square Punnett square if you simplify it. It actually just becomes a 4. square. There's only 4 different options here. So we know that all offspring are going to get this combination of alleles from their mother, and then the only question is which of the sperm that their father can make are going to come in. So capital Y, lowercase y, capital Q, lowercase q, or capital Y, lowercase y, lowercase q, or lowercase y, capital Q, lowercase q, or lowercase y, lowercase q, lowercase q. Now, what are the phenotypes of these organisms? I didn't write out what these genes actually represent, but it's just an example, so I'll just say that this one is Y dominant and Q dominant. Whatever trait is coded for here, it's getting both dominant traits in this case. This one is Y dominant, Q recessive. This one is Y recessive and Q dominant. And this one is Y recessive and Q recessive. And there's an equal number of each combination. So what is this? This is a 1 to 1 to 1 to 1 genotypic ratio. And a 1 to 1 to 1 phenotypic ratio as well, because there's no duplication of phenotypes here, right? Dominant, 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 recessive, recessive, dominant, recessive, recessive. So you get a 1 to 1 to 1 there. And you can do this with some of the simpler genetics problems we've looked at as well, kind of simplifying the Punnett square to make your life a little bit easier. Here's my, let's just get rid of that, pop a new layer on there. So let's say that you have a parent with these alleles, and we're doing a cross with a parent with these alleles. Well, again, we're just mapping out what kind of gametes they can make. So the father here can make 2 different kinds of sperm cells, but since the mother is homozygous recessive in this case, she can really only make 1 kind of egg cell. So instead of doing the full, like normally this is what we learn to do at the beginning, right? Do the 4 square Punnett square every time, and then you would get this, right? Your typical distribution here. But that gives you a 2 to 2 ratio, which we then reduce to 1 to 1. If you think about it ahead of time, though, you can just see that there's only 1 kind of egg cell that this mother can make. So you can do that reduction ahead of time and save yourself a little bit of drawing. You could say there's 1 possibility where a dominant sperm will fertilize a recessive carrying egg. And then there's another possibility where a sperm carrying a recessive allele will fertilize a egg carrying a recessive allele here. So you get the same ratio, 1 to 1. So sometimes when you guys are answering questions, you might be able to use this in order to save yourself some time, but only do that if you're confident. You can only do this with homozygotes. I guess that should be the thing. This individual is homozygous recessive. And because they are homozygous, you can collapse the possibilities. You can make simplified Punnett squares as a result.